Good day, everyone. Today we will be discussing about the enzymes, the biological catalyst, and here are the presenters.
Hi, my name is Bianca Barangan.
My name is Jaren Chris Lyle Burgos.
My name is Alexis Angela Cabrica.
I'm Denver Josh Cadiz.
I'm Kylerine Clemente.
And I'm Neil Justin Vitalvero.
So before we start our discussion, let us know first the definition of an enzyme. An enzyme is a compound, usually a protein that acts as a catalyst for biochemical reaction. Enzymes cause cellular reactions to occur millions of times faster than corresponding uncatalyzed reaction, meaning it typically speeds up a reaction or increases the reaction rate. As you can see, as catalysts, enzymes are not consumed during the reaction but merely help the reaction occur rapidly. Most enzymes are globular proteins. Some are simple proteins consisting of entirely amino acid chains. Others are conjugated proteins, additional chemical components. Enzymes differ from non-biochemical laboratory catalysts, not only size being much larger, but also in that their activity is usually regulated by other substances present in the cell in which they are found. So carbohydrates, lipids, bound metal ions, and other organic compounds. So to further elaborate about the enzymes, we have Neil.
Now let us proceed to the structures of enzymes. Enzymes can be divided into two general structural classes, simple enzymes and conjugated enzymes. A simple enzyme is an enzyme composed only by protein, while conjugated enzyme is an enzyme that has non-protein part in addition to a protein part. The conjugated enzymes consist of apoenzymes, cofactors, and holoenzymes. Apoenzymes need cofactors because it provides additional chemically reactive functional groups beside those present in the amino acid chains of apoenzyme. The term holoenzyme is often used to designate the biologically active combined apoenzymes, cofactors, and entity. Here in the structures, we can see that holoenzymes is produced by combining apoenzymes and cofactors. Aside from that, we have coenzymes, which is the secondary category of cofactors. It is small organic molecules that serve as cofactors in conjugated enzymes. Now that we know the structures of enzymes, let us proceed to the nomenclature of enzymes presented to you by Alexis.
So now let us proceed on how we name enzymes or the nomenclature of enzymes. Enzymes are typically named using a systematic nomenclature system developed by the International Union of Biochemistry and Molecular Biology. This system is based on the type of reaction catalyzed by the enzyme and the substrate upon which it acts. Enzymes' names are typically composed of two parts, and the first part describes the type of reaction catalyzed by the enzyme, and the second part describes the substrate upon which it acts. The first part of the name is usually derived from the name of the substrate, with the suffix ase or ase added to indicate that it is an enzyme. For example, the enzyme that catalyzes the hydrolysis of sucrose is called sucrase. In some cases, enzymes are named after the scientists who discovered or characterized them. So for example, the enzyme named Taq polymerase, which is widely used in molecular biology, is named after the bacterium Thermus aquaticus from which it was isolated. So in recent years, there has been an effort to standardize enzyme names by assigning them uniquely in numerical identifiers based on their catalytic function, known as Enzyme Commission or EC. These numbers provide a more specific and precise way of identifying enzymes and their catalytic functions. The EC number is composed of 4 numbers separated by dots, and each numbers represent a different level of classification. So the first numbers indicate the type of reaction catalyzed by the enzyme, such as oxidoreductases, transferases, or hydrolases. The second number provides more information about the substrate or functional group that is acted upon the enzyme. The third number specify the nature of chemical reactions such as oxidation reduction, hydrolysis, transfer of functional group, and etc. The fourth number serves as a serial number to differentiate enzymes with similar functions.
Classification of enzymes. Enzymes may be classified according to the type of reaction that they catalyze. The six classes are as follows. Number 1, oxidoreductases are enzymes that catalyze oxidation reduction. Lactate dehydrogenase is an oxidoreductase that removes hydrogen from a molecule of lactate. Other subclasses of the oxidoreductases include oxidases and reductases. Number 2 is transferase. Transferase is an enzyme that catalyzes the transfer of a functional group from one molecule to another. Two major subtypes of transferases are transaminase and kinases. Number 3 is hydrolysis. Catalyze hydrolysis reaction, that is, the addition of a water molecule to bond resulting in bond breakage. These reactions are important in the digest process. For example, glycosidases, such as alpha amylase, catalyze the hydrolysis glycosylic bonds between monosaccharides in a polysaccharide, and protease catalyze the hydrolysis of peptide bonds in protein to release amino acids. Lipase catalyze the hydrolysis of the ester bonds in triglycerides. Number 4, lyases. Catalyze the addition of a group to a double bond or the removal of a group to form a double bond. Fumarase is an example of an IVase. In the citric acid cycle, fumarase catalyzes the addition of a water molecule to double bond of the substrate fumarate. The product is malate. Number 5 is tomerases. Rearrange the functional groups within a molecule and catalyze the conversion of one isomer into another. For example, phosphoglycerate mutase converts one structural isomers 3-phosphoglycerate into another 2-phosphoglycerate. The last one is ligases. Are enzymes that catalyze a reaction in which a carbon to carbon, carbon to sulfate, carbon to oxygen, or carbon to nitrogen bond is made or broken. This accompanied by an ATP or ADP interconversion. For example, DNA ligase catalyzes the joining of the hydroxyl group of a nucleotide in a DNA strand with the phosphoryl group of the adjacent nucleotide to form a phosphester bond.
So moving on to our next topic, we have the models of enzyme action. So we have here the enzyme active site. So what is the enzyme active site? It is the small portion of an enzyme molecule called the active site. It participates in the interaction with the substrate or substrates during a reaction. The active site is the relatively small part of an enzyme structure that is usually involved in catalysis. It is the place where substrate binds to enzyme. It is formed due to folding and bending of the protein, usually Kravik-like location and or crevice-like location in the enzyme. Some enzymes have more than one active site. Moreover, the active enzyme or the active site in an enzyme is a 3D entity formed by groups that come from different parts of protein chains. These groups are brought together by the folding and bending. The active site is usually, just like what I said, a crevice-like location in the enzyme. Next, we have the enzyme substrate complex. So it is the catalysts offer an alternative pathway with lower activation energy through which a reaction can occur. In enzyme-controlled reactions, this alternative pathway involves the formation of an enzyme substrate complex as an intermediate species in the reaction. So it is needed for the activity of enzyme. In
Intermediate reaction species form when substrate binds with the active site. Orientation and proximity is favorable in fast reaction. Okay. Within the enzyme substrate complex, the substrate encounters more favorable reaction conditions than if it were free. So the result is faster formation of product. So we have two models for substrate binding to enzyme, and these are the lock and key model and the induced fit model. So first, let us discuss about the lock and key model. To account for the highly specific way an enzyme recognizes a substrate and binds it to the active site, researchers have proposed several models, and the simplest of these models is the lock and key model. So as you can see on our screen, this is the illustration of the lock and key model. So enzyme has a predetermined shape for the active site, and only substrate of specific shape can bind with the active site. Okay. Moving on to the next model, we have induced fit model. The induced fit model is used for this type of situation. The induced fit model allows for small changes in the shape or geometry of the active site of an enzyme to accommodate a substrate. A good analogy is the changes. Okay. Allow small change in space to accommodate substrate. So for the experimental evidence indicates that many enzymes have flexibility in their shapes. They are not rigid and static. There is constant change in their shape. So moving on to the next topic, we have the enzyme specificity.
Let's discuss the enzyme specificity. So what is enzyme specificity? It is the ability of an enzyme to bind only one or a very few substrates. To illustrate the specificity of enzymes, consider the following reactions. The enzyme urease catalyzes the hydrolysis of urea to carbon dioxide and ammonia as follows. So the number one type of enzyme specificity is the absolute specificity. It is an enzyme that catalyzes the reaction of only one substrate. It has an amino acid tRNA that synthetases exhibit absolute specificity. Each must attach to the correct amino acid to the correct transfer RNA molecule. If the wrong amino acid is attached to the transfer RNA, it may be mistakenly added to a peptide chain, producing a non-functional protein. So the next type of enzyme specificity is the group specificity. It is an enzyme that catalyzes reactions involving similar molecules containing the same functional group that has group specificity. Hexokinase is a group specific that catalyzes the addition of phosphoryl group to the hexose sugar glucose in the first step of glycolysis. Hexokinase can also add up a phosphoryl group to several other 6-carbon sugars. The next type of specificity is called the linkage specificity. It is an enzyme that catalyzes the formation or breakage of only certain bonds in a molecule that has linkage specificity. Proteases such as trypsin, chymotrypsin, and elastase are enzymes that selectively hydrolyze peptide bonds. Thus, these enzymes are linkage specific. So the last one of our class of enzyme specificity is called the stereochemical specificity. An enzyme that can distinguish one enantiomer from the other has stereochemical specificity. Most of the enzymes of the human body show stereochemical specificity because we use only D sugars and L amino acids.
The enzymes involved in digestion and metabolism recognize only those particular isomers, stereoisomers. Going to discuss the regulation of enzyme activity. So what is regulation of enzyme activity? So within a cell, it is a necessity for many reasons. It is the control of the rate of reaction catalyzed by an enzyme by some effector or by alteration of some condition. Allosteric enzymes. These are enzymes that have more than a single binding site called allosteric enzymes. They have active sites that can be altered by the binding of small molecules called effector molecules. So there are two processes involving allosteric enzyme. Number one is the positive allosterism. Effector binding converts the active site to an active configuration. Number two, negative allosterism. Effector binding converts the active site to an inactive configuration. So to compare, positive allosterism, they turn active site to an active configuration. However, the negative allosterism converts the active site to an inactive configuration. In either case, binding of the effector molecules regulates enzyme activity by determining whether it will be active or inactive. So as you can see in the diagram, allosteric activation becomes the active site becomes available to the substrates when a molecule binds to a different site on the enzyme. On the other hand, allosteric deactivation or negative allosterism, the active site becomes unavailable to the substrate when a regulatory molecule binds to a different site on the enzyme. The second regulation of enzyme activity is the feedback inhibition. Allosteric enzymes are the basis for feedback inhibition of biochemical pathways. It is a process in which activation or inhibition of the first reaction in a reaction sequence is controlled by a product of the reaction sequence. So this occurs when an end product of a pathway accumulates as the metabolic demand for it declines. The final product in the chain may inhibit the activity of the first enzyme. When the concentration of the final product is low, all of the reactions proceed rapidly. As the concentration increases, however, enzyme 1 becomes inhibited and eventually stops. This end product in turn binds to the regulatory enzyme at the start of the pathway and decreases its activity. The greater the end product levels, the greater the inhibition of enzyme activity. The next one would be the proenzymes. It's another means of regulating enzyme activity. It involves the production of enzyme in an inactive form called proenzyme. The proenzyme is converted by proteolysis or the hydrolysis of the protein to the active form when it has reached the site of its activity. These are inactive form of enzyme, the proenzymes, which can be activated by removing a small part on their polypeptide chain. They are mostly the digestive enzymes and blood clotting enzymes. They are products of entirely different pathways of reaction within the cell. They are also compounds produced outside the cell, such as hormones. So as you can see in the diagram, you have here the activation of the asparase. Okay, so the next one will be presented by Denver Cadiz. He'll be discussing about protein modification and isoenzymes.
Let's proceed to protein modification. The modification is usually a change in primary structure. So the protein modification is typically addition of functional group covalently bound to apoenzyme. One of the best example is enzyme modification is by phosphorylation, and a phosphate group is often bonded to a tyrosine residues, and protein are often modified through a combination of post-translational cleavage and the additional of functional group through a stepwise mechanism of protein maturation or activation. One example of this protein modification is the picture that uh, you're looking at, and this process is affecting the enzyme activity, covalently modifying it. So you can see the inactivated zymogen and the activated enzyme, which is cleavage or remove a part of it, so the enzyme can be activated. And isoenzyme. Isoenzyme occurs when the same enzyme appears in different form in different tissues, or we can say in different version of the same enzyme that catalyze the same biochemical reaction. An enzyme that occurs in multiple forms, and each enzyme the same reaction. Other definition of isoenzyme is that perform the same function but have different combination of subunits and thus different quaternary structures. Example of isoenzyme is the H and M chains, which is the tetramer enzyme, and H4 is present in predominantly in heart muscle, and M4 is present in predominantly in the liver and skeletal muscle. There are also examples like H3M, H2M2, and HM3. And H4 is allosterically inhibited by high level of pyruvate, while M is not, and H4 is serum correlates with the severity of heart attack. Example of this is like lactate dehydrogenase, where we can call it LDH and catalyzes the oxidation of lactate to pyruvate.
So that sums up all the topics regarding about the enzymes. And here we have the list of reference of the book we gathered from the library. And on behalf of our presenters, we would like to thank you for watching, and we hope that you learned something new today.